Thank you very much. And thank you for the opportunity to tell you that proteases would be the key food ingredients to allow the green transition. Leif Nielsen already told you that food need to be appealing and attractive to eat, so we need to have food which would induce an indulgent pleasure when we are eating it. And as the sustainable food production is basically that we will have to say goodbye to animals. That means we will also have to say goodbye to meat, milk, and eggs. And we will have to say goodbye to animal fats. That might be less of a problem. It could give some gastronomy problem in some dishes, but we might be better off without it. But the big problem is that we need to say goodbye to animal proteins. Animal proteins are high quality proteins, so when we are going to substitute it with plant-based proteins, we will need to refine the plant proteins. But probably more important is that we will have to say goodbye to centuries of gastronomy where we have developed the functionality of the animal proteins. The foods you see on these pictures are not necessarily telling you that you are going out there to eat an animal for getting it. But the functionality and what gives the pleasure of those products comes from animal proteins. And therefore, we need to develop methods to obtain similar functionality from the plant proteins. We have been running a project for the last 5 years called Proteases. That's a collaboration between 5 companies and 2 different universities. And we have been developing methods to use bioinformatics to analyze the protein sequences in whatever proteins. And in this project, we have been focusing on plant proteins, and we can continue that route also in the future. And we have been able to develop algorithms which would allow us to predict preservative functionality like antioxidant, antimicrobials, and also emulsifying capacity. So 2 key properties of the food ingredients we use. And we have been able to do that from potato protein and from seaweed proteins. And with the same algorithms, we can do it on a number of other proteins. The project has been very successful, and we are ending this year, and the number of papers coming out of it would be probably around 20. More than 10 have already been out, and we are writing on many more. So the outcome of this can help facilitate the getting texture and shelf life into a number of food products. In the food we know already, proteinases have been a key food ingredient for a long time. The first industrial enzyme was derived from, that was animal rennet. That was developed 150 years ago. And that's a very specific protease extracted from calf stomachs, and it's useful for processing milk into cheese. This enzyme is useless for plant proteins because it has only this very specific property, but we can use it as an inspiration for what we need to do on plant proteins. Also, starter cultures are well known, and in the food we know, like fermented sausages, fermented milks, starter cultures are important, and in this process, the proteinases of starter cultures are very important for their functionality. So we can use the well-known animal-based products as an inspiration for what we want to do. Those products arose not by the brilliance of scientists knowing that we need proteinases. They are the outcome of hundreds of experience in gastronomy. And then the food has inspired the scientists to study what's actually going on. Now I think we need to pay back to this, that the scientists need to accelerate the work of predicting what is needed to get interesting product out of plant proteins. So proteinases specific for plant proteins will allow us to create some of the indulgence and pleasures we are used to get from plant proteins. I would say lactic acid bacteria could be one of the sources we would use for the proteins. It's not going to be the same lactic acid bacteria as we use in the dairy industry or in the meat industry, because the proteinases of lactic acid bacteria are very selective and very specific. So they have the ability to more or less hydrolyze any bond, but they don't hydrolyze everything. So the balance between getting the specific hydrolysis without over-digesting and spoilage is one of the things we can get out of lactic acid bacteria, but we need to find the right ones. And this is what we do in the research we are doing here. And I would like to acknowledge two of the PhD students, Lisa and Ivy, which would be presenting also in this afternoon. We have had several students, and I have also colleagues which are working in this field. So we would be very much interested in discussing with you in the afternoon what we can do in this field. But the take-home message is, as Leif told you, that you need to have pleasure in eating food, and the key in the future would be the food ingredients, and the key food ingredients would be the enzymes of the class of proteinases. Thank you very much.